I'm supposed to scare you. This is about fear, right? And you should be really afraid, but not for the reasons why you think you should be. You should be really afraid that, if we stick up the first slide on this thing, there we go, that you're missing out. Because if you spend this week thinking about Iraq and thinking about Bush and thinking about the stock market, you're going to miss one of the greatest adventures that we've ever been on. And this is what this adventure is really about. This is crystallized DNA. Every life form on this planet, every insect, every bacteria, every plant, every animal, every human, every politician is coded in that stuff. And if you want to take a single crystal of DNA, it looks like that. And we're just beginning to understand this stuff. And this is the single most exciting adventure that we have ever been on. It's the single greatest mapping project we've ever been on. If you think that the mapping of America has made a difference, or landing on the moon, or this other stuff, it's the map of ourselves and the map of every plant and every insect and every bacteria that really makes a difference. And it's beginning to tell us a lot about evolution. It turns out that what this stuff is, and Richard Dawkins has written about this, is this is really a river out of Eden. So the 3.2 billion base pairs inside each of your cells is really a history of where you've been for the past billion years. And we can start dating things, and we can start changing medicine and archaeology. It turns out that if you take the human species about 700 years ago, white Europeans diverged from black Africans in a very significant way. White Europeans were subject to the plague. And when they were subject to the plague, most people didn't survive, but those who survived had a mutation on the CCR5 receptor. And that mutation was passed on to their kids because they're the ones who survived, so there was a great deal of population pressure. In Africa, because you didn't have these cities, you didn't have that CCR5 population pressure mutation. We can date it to 700 years ago. That is one of the reasons why AIDS is raging across Africa as fast as it is, and not as fast across Europe. And we're beginning to find these little things for malaria, for sickle cell, for cancers. And in the measure that we map ourselves, this is the single greatest adventure that we'll ever be on. And this Friday, I want you to pull out a really good bottle of wine, and I want you to toast these two people. Because this Friday, 50 years ago, Watson and Crick found the structure of DNA. And that is almost as important a date as the 12th of February when we first mapped ourselves. But anyway, we'll get to that. I thought we'd talk about the new zoo. So all you guys have heard about DNA, all the stuff that DNA does, but some of the stuff we're discovering is kind of nifty because this turns out to be the single most abundant species on the planet. If you think you're successful or cockroaches are successful, it turns out that there's 10 trillion Pleurococcus sitting out there. And we didn't know that Pleurococcus was out there, which is part of the reason why this all-species mapping exhibit or mapping project is so important. Because we're just beginning to learn where we came from and what we are. And we're finding amoebas like this. This is an amoeba dubia. The amoeba dubia doesn't look like much, except that each of you has about 3.2 billion letters, which is what makes you you, as far as gene code inside each of your cells. And this little amoeba, which sits in water in hundreds and millions and billions, turns out to have 620 billion base pairs of gene code inside it. So this little thingamajig has a genome that's 200 times the size of yours. And if you're thinking of efficient information storage mechanisms, it may not turn out to be chips. It may turn out to be something that looks a little like that amoeba. And again, we're learning from life and how life works. This funky little thing. People didn't used to think that it was worth taking samples out of nuclear reactors because it was dangerous. And of course, nothing lived there. And then finally, somebody picked up a microscope and looked at the water that was sitting next to the cores. And sitting next to that water in the cores was this little Deinococcus radiodurans doing a backstroke, having its chromosomes blown apart every day, 6-7 times, restitching them, living in about 200 times the radiation that would kill you. And by now, you should be getting a hint as to how diverse and how important and how interesting this journey into life is. And how many different life forms there are. And how there can be different life forms living in very different places, maybe even outside of this planet. Because if you can live in radiation that looks like this, that brings up a whole series of interesting questions. This little thingamajig. We didn't know this thingamajob existed. We should have known that this existed because this is the only bacteria that you can see to the naked eye. So this thing is 0.75 millimeters. It lives in a deep trench off the coast of Namibia. And what you're looking at with this namibiensis is the biggest bacteria we've ever seen. So it's about the size of a little period on a sentence. Again, we didn't know this thing was there 3 years ago. We're just beginning this journey of life in the new zoo. This is a really odd one. This is ferroplasma. The reason why ferroplasma is interesting is because it eats iron, lives inside the equivalent of battery acid, and excretes sulfuric acid. So when you think of odd life forms, when you think of what it takes to live, it turns out this is a very efficient life form, and they call it an archaea. Archaea means the ancient ones. And the reason why they're ancient is because this thing came up when this planet was covered by things like sulfuric acid and batteries, and it was eating iron when the Earth was part of a melted core. So it's not just dogs and cats and whales and dolphins that you should be aware of and interested in this little journey. Your fear should be that you are not, that you're paying attention to stuff which is temporal. I mean, George Bush, yeah, he's going to be gone, all right? Life isn't. Whether the humans survive or don't survive, these things are going to be living on this planet or other planets. And it's just beginning to understand this code of DNA that's really the most exciting intellectual adventure that we've ever been on. And you can do strange things with this stuff. This is a baby gar. Conservation group gets together, tries to figure out how to breed an animal that's almost extinct. They can't do it naturally, so what they do with this thing is they take a spoon, take some cells out of an adult gar's mouth, code, take the cells from that and insert it into a fertilized cow's egg. Reprogram cow's egg, different gene code. When you do that, the cow gives birth to a gar. We are now experimenting with bongos, pandas, elands, Sumatran tigers, and the Australians, bless their hearts, are playing with these things. Now, the last of these things died in September 1936. These are Tasmanian tigers. The last known one died at the Hobart Zoo. But it turns out that as we learn more about gene code and how to reprogram species, we may be able to close the gene gaps in deteriorated DNA. And when we learn how to close the gene gaps, then we can put a full string of DNA together. And if we do that and insert this into a fertilized wolf's egg, we may give birth to an animal that hasn't walked the earth since 1936. And then you can start going back further and you can start thinking about dodos.